Good morning. Good morning. Good morning. Good morning. Welcome. I hope you're dry and you remember what the sun looks like, because after a whole long period of having the sun and no rain for months and months and months, we now have this, which is great. We need it. So, welcome. Today, we're going to be talking about respiration and diffusion and some structures that permit the vast invertebrate fauna of the world to respire. Let's take a quick second to acknowledge a complete victory here by all of you. Well done. With the word search, and we'll clear those. Don't lose your annotation tools, because of course we are going to be using them again as we go. So, previously on Zoology 3700, we were talking about, we made a transition from thinking about last week, from thinking about life history strategies that had a lot to do with sex and the evolution and maintenance of sex and different kinds of sex across the animal world. And we talked on Monday about some of the specific types of mating rituals, the rating rituals, and different pressures that might maintain those through evolutionary time. And we then made a transition to thinking about insect or arthropod development and holometabolic, hemimetabolic, ametabolist development. And then we started talking about metamorphosis and molting. And that lets us think about, we were talking about hormonal control of that, and that brings us into thinking about the body and the body wall. That leads me to think about respiration. And so respiration is where we're going to sit and breathe here for the next couple of lectures, for today and for next Monday. And what I'd like to spend today's time talking about is kind of going back to some first principles that are, I'm going to hazard a guess, likely a review for a lot of you, to talk about diffusion, to talk about Fick's law, and we'll talk about different strategies that invertebrates have across the Dunn et al. Phylogeny, across this roadmap, this phylogenetic roadmap that we have for animal life to deal with problems associated with respiration. And we'll continue with that on Monday following the weekend. So, my learning objectives for you, for us, for this period, they're kind of two things. One of them is to be able to describe components of Fick's law, which is displayed right there, and then to interact with examples from the natural world and interpret how this law then affects invertebrate respiration and different strategies that invertebrates have for respiring. And then those strategies and organs are used for respiring. So this is one of the parts that we're going to talk about Fick's law. This is Fick right here. He was a German who lived in the late mid-19th century, who made some really cogent and useful observations about the physical transfer of particles through and across membranes. So his basic, the diffusion rate for an element, for, let's think about oxygen if we're going to talk about respiration, the diffusion rate here being a function of the a product of the diffusion coefficient, the concentration gradient, the area over which the reaction is taking place, and the barrier thickness. So the emphasis here is not on numeric literacy to be able to derive this equation. If you're going to see this equation in any of the case studies or in the final exam, you would see it. It wouldn't be a question of reproduce this for me. It would be, let's think about it graphically, let's think about it visually, or let's think about consequences of this relationship, and you would be presented with that relationship. But let's talk a little bit about it. So the first heads up, I suppose, is that when we're talking about respiration here, the respiration we're not talking about biochemical respiration. We're talking about physiological respiration. So the movement of oxygen from the outside environment to cells within tissues, and then the removal of carbon dioxide in the opposite direction into the surrounding environment. So respiration, gas exchange, so the intake of oxygen and the release of carbon dioxide. And that kind of cellular respiration uses oxygen and then releases many, many more times more energy than does anaerobic cellular respiration. And across the Dun phylogeny, the vast morass, the great diversity of the invertebrate strategies, the animal strategies that are invertebrate strategies, their strategies for respiration, for dealing with varying abiotic conditions, varying concentrations of oxygen associated with temperature and salinity, etc., leads to numerous strategies associated with morphology, physiology, habitat selection, so behavior and evolution. And these can extend from cutaneous respiration, cutaneous, your skin, and then different specialized structure. And we'll finish today talking about some of these specialized structures. But what I would like to do to start is to take a look at this video here.
The blood needs to be taken away from the exchange area quickly so that you maintain the concentration gradient between the two areas. Diffusion happens quickly when there is lots of gas in one place and not very much in the other. This is called...
The break in the video is me just letting people into the waiting room because, of course, computers can't do two things at once.
The concentration gradient. So as soon as oxygen enters the blood, it needs to be transported away so that more can enter the blood next time you breathe in. Being moist also helps dissolve the gases and speed up exchange.
So I'm hoping that as you were listening to this, that there is some of the, not just the voice coming through, but there's some tantalizing late 80s, early mid 80s synthesizer in the background there that is just beautiful.
The blood.
Yeah. So if we're going to talk about cellular or physiological respiration, gas exchange, we're going to be talking about diffusion, right? And so how do gases move? How do gases move? They move via diffusion. And you're familiar with this. And you're familiar, we've talked about it certainly in our shared time together in 2700, but you've talked about it throughout your training as a biologist and across various parts of your career, where you see basically the setup, a concentration gradient shown here at the start in the different colors on the left-hand side between the filled in dark circles and then the filled in orange circles that kind of diffuse across from higher concentration to lower concentration gradient. And when we're talking about cellular respiration, we're certainly invoking some kind of a membrane across which that transport is happening, or that movement is happening. And so here on the right-hand side, we have time going from left to the right, and we see the concentration of something, perhaps it's oxygen in the outside environment, aqueous oxygen in water, across the skin and moving into the body, or in this case, into the cell, across the phospholipid bilayer. With here your spaghetti balls. So Fick's law, throw in the chat, I've said a couple of times that I'm going through this presuming that you've bumped into this before, but tell me if there's anyone for whom Fick is a new character, a new person in the play of life, let me know. Fick's law is essentially the equation that's describing diffusion. New, not new. Okay. All right. That's a 50. This is great. 50-50 split. So we'll introduce and we'll review. But this is the equation that I showed before that we're going to spend some more time with that's describing diffusion. And it's used to solve for this parameter, this diffusion coefficient. And it varies across media. So it's greater in air compared to water, and it varies by temperature. There's a whole bunch. So the abiotic, we're going to focus on some biotic variables a lot today when we speak specifically about organs and structures and strategies. But we're going to also fold in some of the abiotic variants that can affect these diffusion and respiration. particularly temperature, and what else might we talk about? Salinity, perhaps.
According to Fick's law, the rate of gas transfer across a tissue plane or membrane is directly proportional to the difference in partial pressures of the gas on the two sides of the membrane and the membrane's diffusing capacity.
Yeah.
The diffusing capacity of a membrane is dependent on several components: tissue, plane, or membrane area, tissue thickness, solubility of the gas, and the molecular weight of the gas. And now, if we substitute the equation for diffusing capacity back into Fick's law, we get this equation.
All right, so depending on how reduced or how to back kind of in the rear view mirror towards first principles we're looking at, we've got two different expressions of Fick's law. And I'm coming back here to the one that we talked about at the start on the blue box to essentially emphasize, I suppose, the elegance of this equation, or the simplicity of this reaction. So we have, first of all, the things to consider from the perspective of the invertebrate zoologist. We have that the fact that diffusion is enhanced if there's a greater surface area. And you know this. You know this from the equation if you want to be reductionist, but you know it from your history, our shared history together in Zoology 2700 and in other courses where you've seen different adaptations to increase the surface area and increase the site over which these actions or reactions can occur. So increasing the area increases the diffusion. Diffusion is enhanced if the barrier thickness is thinner. And so again, you've seen different examples of this through your training and through your life experience as a zoologist already, that we don't see respiration generally occurring across thick cutaneous barriers. We see them occurring across thin barriers. And so even with the trachea, which we'll come to later on, that is a chitinous, that's part of the exoskeleton. Remember the trachea leading to the tracheoles, leading to diffusion and respiration for insects. That is across chitin, but what you're seeing there is not the hard chitinous exoskeleton, the elytra of that beetle. You're seeing a super reduced, so a reduction in barrier thickness to enhance diffusion. We similarly see an enhancement to diffusion if there's a greater concentration gradient across the membrane. So here we can talk about we'll talk about boundary layers and different behavioral and morphological strategies to enhance this gradient. But let's, first of all, so we've talked about it. So you've seen a couple of videos with some cheesy synthesizers from the 80s in the background. We've called out components of this equation. Let's look at it in terms of elements. How would you see this graphically? Because that's one of the main kind of learning outcomes of the numeric literacy that we have for this course, is to be able to interact with phylogenies and with figures to interpret and synthesize information about the invertebrate diversity. So in each of these three different plots, we have a y-axis that's the diffusion rate. So how is diffusion rate affected by barrier thickness, by area, and by concentration gradient? Fine, yeah, do it. Go ahead. Show me what you would anticipate there, please. I like it. I like keen annotation. Oh, this is a great start. There's like lots of caffeine pumping through veins and shared experience. This is amazing. This is my round of applause for you. All right, thanks everybody. Let me pull those, clear those. So I'm getting up my white erase and we're drawing all those, but I'm saying, let's have a look at, because this is exactly, you're right on the right track. So verbally, the back of the napkin expression of this component of Fick's law is that as barrier thickness is reduced, that diffusion is enhanced. And you could talk about it directionally and say an increasing thickness is a reduced efficiency in diffusion. The opposite direction for area, that if you're talking about the membrane being the skin or some other respiratory surface, that if you increase the size of that, if you've got a great big sail, or increased folds like gills, then you're going to see an increase in the rate of diffusion. And then, of course, there's the concentration gradient, that as you increase that concentration gradient, the diffusion efficient, the passage of that molecule is going to increase. So well done. That's our thinking about Fick. We're going to, I'm going to, Caramel, who's not in the room right now, but Caramel here. Oh, jittery computer, my apologies. All right. I don't see any specific questions in the chat, so we will bid adieu to Caramel for this moment. And we'll think about so I think it's intuitive, and we will pull in examples to come, thinking about the thickness of the layer, the surface area of the layer. But let's talk about how do you maximize that concentration gradient. And this has to do a little bit with pulling back some of what we talked about in the first week, which was thinking about Reynolds laws and thinking about different kinds of flow, like laminar flow, viscous flow, and turbulent flow. So turbulent being the big organism side, and then viscous flow being much more important and defining to the small things in the world. These elements of viscous or turbulent flow, laminar or turbulent flow, come into play irrespective of size when we're talking about respiration, because we're talking about very small parts of the body. And what I'd like you to do here is to picture this being an invertebrate. Let's picture it as a planaria, as a flatworm, like our little bisected flatworm. And so you're looking at this really simplified, small, flat, and thin invertebrate, kind of rolling, creeping along, maybe on the surface of the rocks in the Aramosa River. And this animal, this invertebrate, is exposed to both turbulent and laminar forces because the current, the medium in which it's sitting, the water, is moving. More turbulent forces are experienced. Those are when we've talked about these forces before in Reynolds numbers, we've talked about movement, and so the farther away they are, or the smaller they are, they're less important, but they are still important from the perspective of respiration. They become more important here farther away, and then the laminar, these viscous layers, they slow down, and we have a problem with a boundary layer. So what I'm going to do is cover up all of the complexity of that area inside of, underneath that curve. And what I want to talk about here is the fact that as we've got how the exchange and the concentration gradient, so the exchange of oxygen across this body wall, is altered depending on how fast the water is moving and on where the water is intersecting with the body. All right. So there is a small amount of layer for whatever object you would put in a moving medium. So in this case, water and a planaria. There is a viscous sublayer, this boundary layer that occurs right beside the body wall, where things slow down and they're not as quick. And we know that boundary layer, so that's the first bullet there. So the velocity decreases exponentially within the boundary layer. That has, I'm thinking, in your intuition, that will have some effect on the concentration gradient. Now, increasing the velocity will push that area down, and that will decrease the boundary layer. And this occurs, like I'm giving you an example here with this planaria on the rocks of the Aramosa River, but this applies to internal fluids as well. So this is this discussion about medium velocity and proximity to the body, or proximity to the exchange surface, affects our understanding or an organism's capacity to maximize maximize that concentration gradient. Okay, so we'll switch graphs here, and we'll think about, we've got the surface of our planaria down here in blue, and it could be another kind of invertebrate, could be a spoonworm, it could be something else. And we've got oxygen concentration here on the top, distance from the surface here, and then what we have plotted are two different lines, two different speeds, two different velocities of that medium. And so this is the consequence. This is how do you increase the concentration gradient from the organism's perspective. You disrupt boundary layers. You seek to reduce the size of that boundary layer. So if there's no movement of the organism, if there's no beating of a flagella or cilia or a respirator or some kind of a flap, a fan, to increase the velocity of the medium, I'm going to stick thinking here about water, we see a really low velocity water, and that you've got as compared to very fast velocity, where we see this tracking of oxygen concentration, and that we see a boundary layer that for slow velocity extends up to a millimeter away from the body surface, and that can be driven down to almost half that just by increasing the speed of the water. And this is advantageous because this then, with slow velocity, then we've got a reduced concentration gradient, so reduced diffusion, so reduced respiration. Increased velocity, increased concentration gradient, increased diffusion, increased capacity to respire, increased capacity to use that oxygen in your muscles and for life as an invertebrate. So the question then is, how would you do that? Well, one of the simple answers is you move around. So what you're looking at, the video that's kind of looping underneath bottom here, is a spoonworm. This is a highly derived Ecurine worm. So this, if you remember in our discussion of the annelids last year in 2700, we talked about spoonworms as being, back in the 20th century when I took the equivalent of this course, this was a phylum of its own. Its segmentation is so secondarily reduced that for a long time we didn't think of them as annelids. And they tend to live in tubes. And those tubes tend to have water that is removed from the current. You can see some of it moving around here. But what the Ecurine does is it uses this peristaltic, this kind of rhythmic action, looking like an intestine that's outside of it's like a Game of Thrones intestine. It's been spilled from its owner, and it's writhing on the ground there. That peristaltic action, in this case, is intentional, and it's an attempt outside of the tube or inside its tube to create water flow, to reduce the boundary layer, to increase the concentration gradient, to allow for greater, more efficient respiration. And you can do this with the whole body, like the peristaltic annelid that we just saw. There are specialized appendages, and certainly cilia. And we've seen, like, in so many different groups, particularly last year when we were talking about protozoans, but even into the ctenophores and some of the larger organisms with cilia. And internal cilia, think about the mollusks and the mollusk gill, and we'll talk about some of that as we go. But let's talk about increasing or accentuating fluid flow using appendages. So we've got a very complicated bit of labeling here. Very, not complicated, it's useful labeling. But we're going to talk about a decapod. So if you're thinking about crustaceans, if you're thinking about eating some lobster, this lobster that is ending its days with you and transferring its energy into you, on its legs, and you remember associated with each one of those legs, there are some of the legs that are associated for feeding, some of the legs are associated for moving, and some of the legs have associations respiratory, have gills on essentially in kind of up in the armpit area. Now there is a specialized region that is this flap right here, the scaphognate, or a gill bailer. And the gill bailer is an adaptation that is shown here on the diagram. So you see we've got from top left to right, and then second row left to right, diagrams where someone has illustrated the movement of the gill bailer outside. So we've got this. These are the gills. These are the filaments of the decapod gill here underneath this kind of M shape, upside down, or let's call it a W then. And in gray, that paddle, that's the gill bailer. And so if you watch it through time and you think of it kind of moving through time, it is creating a power stroke and a recovery stroke like this, where it nearly disappears, and then it pushes again. And so that's that paddling that gill bailer is doing with a recovery period creates not just flow, but a unidirectional flow across the gill surface. So this is, in this case, a specialized elaboration of the exoskeleton that creates water flow, that accentuates the concentration gradient, that accentuates diffusion and respiration. And this is one of a large number of adaptations to try and get around this, what's been called a 1 millimeter rule. So this is the fundamental limit of diffusion. Now back in 2700, early in the term, we talked about why you don't need to be afraid of 1950s horror movie insects, large, giant ants kind of moving through the environment, and you don't need to be afraid of them because if they're breathing in the same way that small ants do, they're collapsing because they can't respire. Because the efficiency of this system outside of 1 millimeter is greatly reduced. And so you would, so the ants, the giant ant threatening your hometown is going to suffocate. And we'll talk about different, and that's because of this limit of diffusion, that this property, this Fick's law really works on a very, very small scale within that 1 millimeter. And there are many adaptations like those gill bailers that try and drive, or adaptations that exist to, including others like increasing surface area and behavioral, and we'll talk about ways to escape this. And those include circulation, which that part, and respiratory pigments and those kinds of things. And we'll talk about those next week. So let me look down. I don't see any calls here. All right. Now let's talk about, so that's kind of the first principles of respiration. Let's talk about some of what evolution has come up with over the billions of years that we've seen life on Earth. What strategies have we seen? What efficient strategies, or efficient enough strategies, has evolution kind of pulled into the foreground? So we talked about cutaneous respiration, and we invoked it, or even dealt with it, in thinking about our planaria moving and hopefully sitting in fast water instead of slow water to increase, drive down the size of the boundary layer to increase the concentration gradient. And we'll talk and continue to talk about gills, these evaginations turned outwards, and then invaginations like trachea or like lungs. And we'll talk about them across of the phylogenetic breadth, or more of some of what you can see here. It will be more, because the very next thing that I'd like to do is to think about, let's think about comparing specialized organs and, say, across the body wall. So cutaneous respiration as being one of the simplest forms. I mean, it is an organ, kind of, but let's think about it from an evolutionary perspective. So the first category that we have here, this is your Dunn phylogeny, of course, and we've got these little parapets poking up here. So echinoderms, our own, the craniates, arthropods, onychophorans, annelids, and mollusks. Really the lineages where we see a vast diversity of respiratory structures present. And then all of the other lineages, there is to some greater or lesser degree evidence for cutaneous respiration. And so what are the respiratory structures we're talking about? These can be the gills, trachea, lung, really specialized things. So what I'd like to see from you in the chat, so this isn't an annotate, but just tell me about it, write it down, show me in the chat. What do you see phylogenetically? What kind of phylogenetic pattern or information do you receive from this comparison of where the structures are present, what you know about the taxa? And so in that what's the trend? Where do you see respiratory structures present? that's great. I see some ideas coming up here. This is wonderful. So the idea that cutaneous respiration is ancestral and respiratory structures are derived. I like that. That's correct. Yes. And then what common elements do we see about? We see a lot of terrestrial organisms. This is true. Yes. Especially, so the example I think about there is the Onychophora, right? This is a phylum that is not found in aquatic or marine systems, but is found in moist terrestrial systems. Yes, that it has happened multiple times, that it would not be a parsimonious decision to say respiratory structures, lungs, evolved one time, and that's it. No. Now think about, so these are all correct, but let's think about from the perspective of where we've just been. We've talked in thinking about life history, so yes, not monophyletic, but think about body size and respiratory organs. Yeah, that's it. That's it. There's a bunch of you jumping. So that's large body size with associated. So larger body size associated with respiratory organs, i.e., there has been a selective pressure that if you're going to escape that 1 millimeter rule, if you're going to be larger, you need to escape the 1 millimeter rule, the diffusive boundary layer. To do that, you need a specialized organ. Let's talk about, that's great, well done. Let's talk about some of these different, and let's start out thinking about the one that we kind of ruled out or we talked about right from the start, the cutaneous respiration. And so there's all sorts of, so we see it in some mollusks, we see it in sponges, nemerteans, nematodes, flatworms, platyhelminth. So this is direct gas exchange across the body wall. It's functional in air or water, but the skin has to be moist. If it's going to happen in the air, the skin has to be moist and highly vascularized. That's kind of thinking about circulation, which we'll come to on next Monday, and then thinking about if this is going to be how you respire, especially if this is the entirety of how you respire. Diffusion is limited by the barrier thickness, so the maximum thickness is about 1 millimeter, and that means, what does that mean in terms of skin thickness? It's going to be very thin, but what does it also mean about body size and body shape? We tend to see with that phylogenetic example, here, for example, so the flatworms, and here is a flatworm example, where we're going to invoke some abiotic conditions here in a second, but lots of these things are very thin. So not just the body wall being thin, but the organism being thin. Now, when we look at, this is a flatworm that we might see in Ontario and around the Grand River watershed, like the Aramosa or Speed River, where we look at a plot of temperature, of water temperature and oxygen consumption. And we see, and I talked about kind of bringing this up, the influence of abiotic factors. This could be, we could do another plot and compare a different set of reactions for salinity, for instance, for brackish organisms living coastally. This one, with temperature, you know intuitively because you probably make coffee or tea at some point in your day, and so you've been probably, as you've taught and learned how to make good coffee or tea, you use colder water, and you're thinking about that largely because of the interaction that water has, and that we've got water being retained more in cold water. But let's think about this in terms of, let's bring in body size again. And I'm going to ask you, for this particular flatworm, what relationship would you predict between body size and oxygen consumption for this flatworm at a single temperature? Annotate away. Well done, all of you. That's great. So I see some nuance and subtlety in some of the plots, And I'm going to take off my glasses and just look at like the big pattern and say this is exactly it, right? Well done. Let me find. I'm going to get my white board marker, clear those, and I'll show you what the data actually looks like. And so it's not as tight, but it is in the direction that you've talked about. And so abiotic features and then biotic features including those as elemental as the size of the body intersecting with how their animals are respiring. Okay, Caramel's further reminder for me to check in and say, let's kind of move through some of these examples. Some of these are definitely review because we talked about them in our time together with 2700. So we'll talk about some new ones and we'll revisit some hits from the past. So water flow across a gill. We see it in annelids. We see it in crustaceans. We see it in mollusks all across the world. Used mostly in marine and some freshwater invertebrates for gas exchange. And these are evaginations of the body surface, which might be exposed, like a gill exposed or enclosed in a cavity. And so they're very thin-walled, so it reduces that layer, the body layer size. So thin wall structure, so it reduces the barrier thickness. And it increases the surface area because we often see this feature become highly folded, like the kind of the arrangement as we talked about with mollusk gills, particularly with the bivalve mollusk, where we see them kind of hung on different kinds of support in arrangements into the water, into the moving water. And then we also saw examples of countercurrent exchange, where the internal beating heart is moving your the fluid containing the respiratory pigment in the opposite direction from the water to increase that concentration gradient. And so we see, for instance, with the bivalve mollusk, we see the things, you know this, that they're hung in this mantle cavity, that they look a lot, I mean, in their name, the ctenidia, these filaments or combs hanging from a central axis, and we see countercurrent exchange. So we've got the water movement, but we also have cilia. So we don't have a peristaltically moving annelid here. We've got more of a static mollusk, but we have the small beating cilia that are accentuating this and creating this current, and the unidirectional countercurrent in then increasing the efficiency of oxygen being extracted. And so, and you've seen that before in this kind of exact kind of a plot. So we've got the blood, the oddly color selected here. The blood flow is actually in black here at the bottom. Water is in red in the opposite direction. So as you move along the gill, the oxygen concentration in the blood increases and it is coming from the water, which decreases. And we see, skipping away from the mollusk example across many other invertebrate groups, this increase in the surface area, so that if we're going to look at these phoronid worms, for example, with their spiraling respiratory surfaces, that we see that if you're going to become a larger phoronid worm, you're going to need a more complicated and more whorls in that to create more surface area. Now, so gills need this constant flow. This has to do with behavioral selection and where they sit, but it also can be then, as we've talked about, created by the cilia. You can move the gill through the water, like if you're a small aquatic invertebrate, or you can move water across the gill. Then that's the ciliary flow. And pump-like action, or that kind of gill flap that we talked about with the decapods. Now, that's a very quick review of something that we spent a lot longer on in 2700, but it's to make space for increasing the number of examples and kind of this appreciation of the diversity of strategies that we see. Who's heard of, show me in the chat, who's heard of plaston respiration before? I find this really cool. Okay. This is amazing. So when we're dealing with insects living in an aquatic environment like this is a picture taken through the side of an aquarium. And it's letting you look at the fact that we are up above. So up here, this is us in the air. Down here, this is the water. And this is a particular beetle that is moving around and still breathing air, even though it is in, its oxygen is being derived from the air and not from the water. So there's no gills happening here. This is tracheal respiration. And that's happening because of the diffusion of oxygen into the this little bubble. You can see this light space here. Now this plastron, or respiratory bubble, is created because the beetle has hairs on its underside that are hydrophobic, that allow the water to be excluded, and for the creation of this little bubble that essentially is functioning as a lung. So it's allowing species to remain underwater indefinitely. So we see that it surfaces, the air bubble is captured beneath the wings, and then we've got kind of water surrounding the bubble, and oxygen is diffusing in, and all of the byproducts of respiration are diffusing out, but we've got the capture in these tiny hydrophobic hairs, which is kind of a lot of times when we're talking about these invertebrate adaptations, I'm like, oh man, wouldn't it be cool if we had this? And this is definitely for me oh man, wouldn't it be cool if we could do this? Now, lungs, and I put it in quotes there. So we've seen lungs. We own them, right? We have them. Our success moving away from the water in the long term is largely about our packed kind of bits of little alveoli and respiratory surfaces there. But we see them across lungs as just a description of these respiratory surfaces packed into a small area. We see them in terrestrial snails or slugs. We see them as respiratory trees and sea cucumbers or echinoderms, and then book lungs. And if you were just to look up lungs in Wikipedia, you'd see, or you would have a little while ago, this sentence, that the lungs are the main organs of respiration system in most terrestrial animals, and to which I hope you would then think, well, actually, most? Really? Most, well, vertebrates, sure, but most animals? The only way we get to most animals is if lung is not that kind of vertebrate example that the Wikipedia showed, but it is in fact a book lung, which you remember is an adaptation of the leg that we've got and the body wall that we see in spiders and arachnids. And so lungs writ large, are often for breathing air. An exception is the sea cucumber, where we see them respiring or pulling oxygen out of the water. But we essentially, it's a word that describes a non-monophyletic thing, an adaptation, but it's moist passageways with thin body walls and circulation. And there's numerous ways of achieving that. And the only way that we would say it's for most animals is if we think about spiders, because spiders, as this study that was talked about in the Washington Post a few years ago, that talked about the world's crushing weight of spiders, where somebody did some back of the napkin calculations and concluded that the spiders of the world weigh about 27 million tons. So most. Now, these book lungs are the stacks of air pockets and tissue that you remember from 2700. We've got them right down here. Highly modified abdominal appendages. And let's take a look at this.
The breathing of the spider is done with the help of the so-called book lungs, or fan tracheae. Some spiders have 2 pairs of book lungs. Other spider species have only 1 pair. Their posterior book lungs have been partially or completely transformed into tracheae. Tracheae and the openings of the book lungs are located on the underside of the abdomen. Gas exchange between oxygen and carbon dioxide takes place in both respiratory organs.
There we go. Now lungs, so book lungs. We also see lungs in some of the lungs in some of the mollusks, and there is a picture of one. This is a banded snail in your dairy bush showing you aerial respiration that some mollusks use in hypoxic or low oxygen conditions. So this is an organ, the pneumostome, and you're going to see it in this video here for the slug, the limace, that's going to open right there. And so it was exposed under the shell in the last one. And the number of pneumatic openings greatly increased. So this is essentially a way of bringing into the interior respiratory, of like gaping, bringing in all sorts of oxygen to increase the efficiency of respiration there for terrestrial snails with the pneumostome. Caramel. Amazing. I don't see any specific questions, but let's talk about, for the last couple of minutes, let's talk about trachea, which we've talked about before. So if we're going to say if we're going to rewrite that Wikipedia page and say what is the main respiratory organ for terrestrial animals, it is this, because based on abundance or diversity, arthropods, insects win, and this is trachea. So we see it in insects, we see it in some spiders, centipedes, millipedes, the little things that run the world use the trachea, and that's all these kind of blind ending white little bags here. And we saw it the other day when we were talking about molting with this example from my backyard of the cicada that had left its larval trachea there as it molted and was beginning to inflate its wings. So these are trachea invaginations of the cuticular exoskeleton we talked about. So reductions in the thickness, and so not having a nice tough carapace like your elytra or your thorax here, but the fact that it's the same material that is creating these highly branched air-filled tubules that move in through the body. So it's reducing, thinking about the diffusion coefficient, we're reducing the thickness of the boundary layer. We're increasing the surface area, so it varies in thickness from micrometers to almost 1 millimeter. That allows then for direct gas exchange between air and tissues. So the spiracle, the opening, leads into the tracheoles that penetrate cells that allow for diffusion of water and the exiting of carbon dioxide. And so there's not generally a circulatory system that's relied on for oxygen transport, so we don't need to worry about giant cicadas threatening our city. So this is more of an open circulatory system with the hemocoel. So we've got the heart that's pumped into arteries, into a hemocoel, into collecting vessels, into kind of tracing through the sinus, the ostia into the heart. And that we see trachea in insects like this, where we've got a spiracle leading into the trachea, into the tracheoles that go directly into individual cells. And with other arthropod lineages, we see gills and the maintenance of gills. Let's talk about the trachea for a second here.
On the underside of its body begins a series of reinforced tubes, adapted from the exoskeleton itself. These tubes open directly to the outside air and draw oxygen into their bodies.
And we've seen, as we talked about in 2700, we've seen changes in that before. So even through linking in our last discussions about developmental changes and major resortings and metamorphosis in the pupa, you can see here the breathing tubes are in blue, the guts are in red, and from day 1 at the top to 13, you see a big reduction in the volume of the gut from caterpillar to preparing to be a slow-moving herbivore to a highly mobile flying adult. And you see the corresponding switch in emphasis from a large gut to a whole bunch of breathing tubes. And then finally, the last thing that I'd like to talk about is writ large, these concentration gradients. We've focused on different strategies and respiratory organs and strategies of the insects or the invertebrates that possess them. But the environments within which they live, those are also changing. And so we see globally dead zones that are shown here in documented hypoxic areas. So coastal areas where the concentration of water is just being driven down, that's creating dead zones where life that requires oxygen can't live. A lot of that is associated with the secondary effects of the increase in phosphates and nitrates that we use terrestrially that flow out into the watershed and flow out into the oceans and then eutrophie, so increase the growth of phytoplankton. We see an increase in biomass and an increase of bacterial growth that drives down oxygen. So that's happening globally, and that's kind of a consistent theme with the Anthropocene. But we also see it happening locally, and we've seen it through time. And it's because of the hypoxic conditions that can be created in lakes that freeze completely over here. So we see reduction in temperature that causes snow and ice to be created, freezing over the lake. That reduces primary production, so the production of oxygen is driven down, and it blocks the air-water interface, so the diffusion of aerial oxygen into the water is reduced. So now we've got in the water the use of oxygen being greater than the production of oxygen, and a lack of replenishment from the air, which means over winter we have potentially hypoxic conditions that are created. So an overwinter hypoxia is a reduction in the dissolved oxygen in part of that water column, following the interference of the ice, the air-water interface. And this can be catastrophic to organisms that live aquatically, and that might be something that you end up, if you're interested in marine and freshwater, if that's the program you're in, it might be something that you can follow up as research projects in the future. This is our Wednesday time, done. Now, thinking about respiration. This is where we've been. Where are we going to next? We're going to talk about different adaptations, more respiration, and thinking about circulation. So a reminder that you continue to not have labs this week, so preparing for next week, you can think about your return to the class experiment, prepare for that lab. Continue to work on your invertebrate R assignment, which is due on the 3rd of October, and the case study for this week will come out on Thursday morning, and you've got until Monday morning to work on. And so my reoccurring reminder that forming study groups where you write questions for each other is probably the best way to prepare for these. So I hope that you're able to do that. Beyond that, I want to say, take care of yourself, take care of the people around you, and I'm going to stop recording and hang around for a section if there's any questions. So